So, good evening. My name is Ankita, and this is the live interaction session for week 8 of the course NPTEL Basics of Biology. And today we are going to go through the basic topics for this week, which is around the human physiology. And particularly this week focused on the process of digestion in quite a lot of quite some extensive details. So we'll go through each of the subtopics under this topic one by one. So first of all, coming back to the basics, the way we recapitulate every week. So when we are trying to understand living organisms, we are going through each and every aspect of their being, where we first started with evolution, moved towards understanding cells, individual biomolecules, then cellular processes, and now we are looking at the physiology. And we're looking, when we say physiology, it basically means the different processes that operate in order to allow an individual or any organism, particularly here we are talking about humans. So these processes that allow a human being to function the way they do. So that is what we are going to look at. And under physiology, you have each of these individual processes, like I just mentioned, starting with the circulatory system, which is basically the reason why an let's say a nutrient or any requisite compound within the body can be transported from one place to another. So kind of if you think about it, if you think about the vascular system which is present in plants, that operates in ensuring that the products, the food products that are synthesized in the leaf, that gets transported or that gets circulated throughout the plant body, or the water which is taken up by the roots is also circulated through and through. So much similar to that, when oxygen is synthesized in the, not synthesized, when it is inhaled during the process of respiration, then that also needs to be circulated. So that is carried out by the circular system. For any movement that an individual will might have to or will have to execute, for that the muscular system comes into play. And then we have the nervous system, which is responsible for bringing about sensations within the individual. The endocrine system for hormonal regulations, the excretory system for removal of any and every unnecessary product from the body. And of course, the topic which is the focus for this week's discussion, which is digestion. So why at all are we bothered with the process of digestion? So like you obviously already know, it stems from the fact that an organism, a living individual, needs nutrition, needs food, and from that food it needs to derive nutrition in order to survive. So nutrition can actually be acquired in a number of different ways, which starts with the mode, where we actually try to understand that how is an organism attaining or obtaining that nutritional value. So in case you think about plants, they are autotrophic because they take it up from they're able to synthesize their own food or their own nutrients by consuming other organic materials. When we say consume, it basically means that they are utilizing the surrounding materials and they have the capacity to synthesize products which are necessary for their own survival. We humans, or any heterotrophic organism for that matter, is incapable of doing so. And as a result, we are heterotrophic, which means that we are dependent on those autotrophic organisms to have the capacity to synthesize food, and then we'll just consume them and obtain nutrition from there, from that source. So we are dependent on such organisms. Apart from that, these are the two broadest ways to classify the mode of nutrition. But apart from that, there are other ways of classifying as well. So that starts with holozoic nutrition. So holozoic nutrition, if you think about it in a very light or very superficial way, it is simply consuming something in whole. So what does it do? It is, or what does an organism that has the holozoic mode of nutrition do? Is that they take up an entire it can either be an entire organism or it can be a huge source of huge food source, and it can be in the form of a whole part of a plant, or it can be a whole part of an animal. And we as humans, since we eat plants, right? We eat fruits and vegetables. Those are parts of plants, and we are consuming an entire part from them. Or if you think about people who follow or who have adapted a non-vegetarian um, food lifestyle, which is where they consume meat or they consume other animals, what they as well are doing are that they are consuming some part of the animal, some part of a fish or other living beings in order to sustain themselves, in order to acquire nutrition from them. So that puts them under the domain of such holozoic nutrition. So examples includes most free living protozoa, humans, and other animals as well, that all fall under this category. Next, we have saprophytic nutrition. So here, what they do is the organisms are able to fulfill their food necessities by, like we also had discussed this in the previous session as well, where we are basically talking about organisms that feed on. And when I say feed on, it means that they're basically trying to derive nutrition, right? So they are consuming or they're feeding on uh, dead, decaying organic matter, it could be some part, some rotten or some dead part of a living organism, and deriving nutrition from it, instead of adapting the holozoic form of life where they consume the organism when it is alive or when it has been caught. Instead, they feed on material that has already decayed, that is dead and decaying, and then they take in the digested food. Now it can also, you can also think of it in this way, that if they have a prey, if they have some organism that they're about to feed on, they are also capable of releasing or secreting some digestive enzymes that would help in breakdown of that potential food material. And then these saprophytes or saprophytic organisms would then be able to consume these parts, and they'll be able to take in this digested food. So what it is effectively doing is that it simplifies or it breaks down the existing food material to make it much simpler, to make it easier to consume, easier to digest, and they're accelerating the entire process of digestion by this um, method. So you can think of it as an adaptive measure, in fact an advantageous adaptive measure that is taken up by a group of relatively less advanced or relatively simple organisms, because the more complex an individual or an organism gets, the more finely regulated would you see these physiological processes. They have a lot of mechanisms that are uh, operating in order to keep a lot of things in check. So that there is a different control that is executed under these circumstances. So that is what we understand about saprophytic nutrition. After that, we have parasitic nutrition. Now, I'm sure each and every one of you is quite familiar with parasites. We have discussed a lot about them during the diversity and classification classes. So basically, a parasite would be any organism that fulfills its requirements, its needs by consuming, not consuming, by depending on another organism. So if you look here, you are seeing that the organism that fulfills the food requirement from the body of another organism. Now, what do we mean by that? What do we mean by fulfilling requirements from another host body? It means that they are depending upon the host, they are latching onto the host, causing some sort of harm to the host, and they are surviving themselves. So it's a different scenario. So if you think about parasites, majorly they are of two different types. Either they can live inside the host body or they could live outside. These are two separate circumstances. When we talk about those that live inside, those are endoparasites, whereas the ones that live outside are ectoparasites. So the internal parasites live within the host body cavity, and more often than not, they are actually quite they're potentially life-threatening, while others live outside. They keep on changing their host. They can get some nutrition from one. They can act as vectors. They can carry something harmful from one host and infect another individual, and there they act as a parasite. So a number of different um, survival strategies are operated by these organisms. And other examples, we have plasmodium, tapeworms, etc. While external ectoparasites or external parasites, majorly they include insects or fleas, where they live on certain animals. Among the fur or hair of certain animals, you would find a lot of parasites. So their lifestyle is quite different. It is quite distinct, and there is an immense dependency that comes in such organisms. So that was about parasitic nutrition. So we talked about nutrition, we talked about the different major classification or major groups under nutrition. And now we are being asked, why do we eat food? So when we talk about food, it basically, it's a necessity, right? It's a requirement for an organism to survive. Now from that food to the process or to the point where you are deriving nutrition from that food is actually the entire process of digestion, right? So like you might have already seen in the lectures as well, that it starts with breaking down the food. When we say breakdown, it basically means you are from a complex material, which can be some sort of a protein, it can be a carb. From there, you're getting some simpler, not necessarily monomeric, but simpler substances. Why I'm saying not necessarily monomeric is that even for a monomer, you need to break it down to each individual atom, right? Or you might have to break it down. You just want to be able to use that particular compound as a source of energy. That is effectively the purpose of food, getting nutrition, deriving a source of energy. That is what the entire process is all about. So here we see that each of the different bonds, each and every bond within a monomer can also have um, like some subtle differences in the amount of energy that would be released if that particular bond is broken down. So here we can see that the CH versus the CO or the OH and the CC bonds, they all have different free energy values that are associated with them. But breakdown of each and every bond releases some amount of energy, which is utilized by the organism. And this process overall is what we understand by the term metabolism. When you are metabolizing a particular compound or product, this is what you're basically doing. Now coming to the purpose of digestion, this has been taken up quite extensively in the lectures as well, where the reference point was a complex food item like a pizza, and each of its individual items, they get digested in different ways by different processes to yield different monomeric products. So for example, bread, it gets broken down into starch, which is a polysaccharide. And when you further subclassify or like when you further break it down, what you effectively get is some sort of a monomer, which you will use as the source of energy, which, like we said, is the entire reason why at all you eat food, why you need to eat food. What does it really help you from a evolutionary point of view, if you think about it that way? Because nothing in nature is, more often than not, nothing in nature is made just because. There is always a reason which is attached to it. And in this case, these individual monomers, which could be utilized as the source of energy, are the example for the same. Sorry. Yeah. Then we come to the second component, which is butter. Majorly, that's a fat, and it forms long chain polymers, effectively giving us fatty acids. And thirdly, we have cheese, which gives us which is sort of a milk protein. It can be considered under the milk protein domain, and from that we get amino acids, which are the monomers that compose them. Now, the process of digestion, as simple as it might seem, it is really not. There is a huge system that is dedicated to it, obviously known as the digestive system. And among that, we can broadly classify that into two groups. One would be the entire gut. When I say gut, I mean the entire alimentary canal, which are each of the individual organs that have individual roles to play in the digestive process, which starts with the mouth. Sorry, starts with the mouth. It from there it goes into each of the different organs, and it ends with the rectum and the anus. On the other hand, if you think about the other set of organs, which actually do not have a mechanical or a very direct role to play, but what they release, what they secrete, which obviously, if you're hearing the term secrete, it means that these are mostly enzymes, right? So these enzymes that are released, what they do is that they basically break down this food further, helping in acquiring nutrition from the food that you're using, all coming back to the same purpose. So that's what we need to understand, that everything that happens within a human body, more often than not, everything that happens is happening for a reason. So that's what we are trying to say here. So this is the anatomy of the digestive system. So it starts from the mouth. Here you have the buccal cavity. From there you have the food pipe, or commonly what is called as the esophagus. From there we have the stomach, the extremely convoluted and tightly what should I say, compacted organ, that is the small intestine, and finally the large intestine, which ends with the rectum and the anus, and that completes the entire digestive tract. It's basically giving you the roadmap that is taken up by each and every food item that you consume. So that's the process that's taken up. Now, coming to each of these different organs, we are obviously going to start, with the buccal cavity. So the buccal cavity, basically, it is the entire cavity. It's your mouth cavity. Let me put it in very common layman terms. Your mouth is basically the buccal cavity, and this is compiled, or you can define its boundaries by the palate at the top, your tongue, your teeth, all together forming the entire cavity where the process of digestion starts at all. So the human mouth consists of vestibule and the oral cavity. What do you understand by the vestibule? The vestibule, on the other hand, is it's the area between your cheeks and your gums. So your gums are hard and your cheeks are on the external. So the space that is between these two, like at the front of the mouth, that is basically the vestibule. On the other hand, the oral or the buccal cavity basically includes the palate, the tongue, the tongue, and the teeth. So mouth is the first passage of food where the piece of food gets digested or broken down into small pieces with the help of teeth, and then it is mixed with saliva. So the tongue manipulates food during chewing and saliva mixing, and this is what it effectively produces is a bolus that goes through the entire tract, through the entire alimentary tract. It moves inwards through the pharynx, finally goes towards the esophagus, and this entire process is altogether called as deglutition or swallowing. So when you're swallowing something, that's the process that comes in. Now, We talked about one specific organ within the buccal cavity, one of the contributing major contributing partners. Um, One of the major contributing partners are the is the tongue. So as you already know, the tongue is the strongest muscle in the human body. So what it is basically doing here is that it is helping in churning of the food products, or the so what you have actually consumed is a much larger food product that needs to be broken down. Now that major role of breaking down is taken, this is where the process of grinding or chewing can actually occur. But at the same time, the other major contributor is actually tongue. If you think about the tongue, if you think about the tongue, it is basically divided into two parts. First is the pharyngeal part, second is the oral part. So the pharyngeal part is the one that is closer, as the name indicates, it is closer to the pharynx, whereas the oral part is much closer to, or it's the side that is towards the mouth. And also what needs to be kept in mind is that the tongue one unique example of one such muscle where it is a different subclassification or subparts of the tongue, starting with the lingual fossil, foramen cecum, sulcus culminaris, different types of papillae. So papillae are basically, they are taste buds. They are the reason why you're able, there are a huge number of receptors present, and they are the reason why you're able to identify one particular flavor, one particular subtype as that subtype and not as anything else. So that comes in later. And we have the valve under the smaller group of papillae point in the tongue of individuals. And altogether, these papillae together give about an idea of what is the flavor profile, or what are the recognition sites within the tongue. So you have salty at the tip, at the tip of the tongue. Then we have sweet, which is behind it. So even suborgan within the buccal cavity, and that actually involves the teeth. I'll just So coming back to what we were discussing. So we were discussing each of the individual parts of the buccal cavity. And under that, first we understood the function of the tongue. And now coming to the second component of it, which are teeth. So teeth are actually considered to be the machinery that is taking up this extremely important job of mechanically breaking down or grinding whatever food particle that each of us take in over the years and do so consistently. And for that, what is well understood and well studied is that each of the different subtypes or each of the different types of teeth are involved in different types of functions. So if you look at the types of teeth, majorly there are four, which are incisors, canines, molars, and premolars. Incisors, which are present right at the front, the rabbit teeth that we commonly understand, they are involved in cutting. So whenever you are taking any food item, say, imagine imagine you're eating an apple, right? So what you bite with, your first teeth, which are at the front, are your incisors, which are taking up the immense responsibility of cutting through something. So they are immensely strong. All of your teeth are strong, no doubt about it, but incisors have a really significant role to play in that as well. Then come the canines. Canines are the sharper, more pointed teeth that you understand are more relevant or more significant for meat eaters, because meat is more elastic in nature. It is not crisp or not as hmm, not as rigid or not as crisp as you would consider an apple to be, right? Meat is more elastic, and therefore, for that, you need sharper tools, for lack of a better word, to cut through such materials. And for that, incisors play that immensely important role. Thirdly, we have molars, which are right at the back of your teeth, which are involved in crushing parts. So after you have cut through some food item, you need a bigger surface area to actually process it, to break it down. That is taken up, that function is majorly taken up by the molars, and they help in crushing and further making it into smaller pieces, which is the process of grinding, is taken up by the premolars. So these are each of the major functions of each of these different types of teeth. Now, where the variation also comes about is in the number of each of these subtypes. So if you consider humans, for example, here you can see the dental formula. This is what is called as the dental formula, because it shows how teeth are arranged. You'll be able to see each of these four subtypes: incisors, canines, premolars, and molars. And usually the formula is 2123 by 2123. What that basically means is that if you consider a human being, this is the up. Sorry, there is some issue with the internet. Uh, what we are also seeing here is that each of these upper and lower palate teeth also they are not the complete set. Find 2, 1, 2, and 3 of each type, so again 2, 1, 2, and 3, and again likewise on this side. So what it is giving you is 8 teeth into 2, so 8 here, giving you 16 on the top and also 16 on the bottom, comprising the entire set of 32 teeth in case of humans. Here you can also see a few more examples, like for herbivores. Even among herbivores, you're seeing a lot of variation of each of these different subtypes, because even though they are herbivores, which means that they feed majorly on plants and plant-based plant products. Despite that, each of these organisms live in different conditions, and therefore they have, sorry, there's a lot of noise. Therefore, they have accessibility or differential accessibility to different types of food items, as a result of which their teeth requirements and their adaptive behavior is also different. So you are able to see slight changes in the dental formula as well. Since we are all omnivores, most humans are omnivores because they feed on both animal and plant products. You will find quite some similarity between us and hedgehog dental formula. So it's about the diet that the organism actually consumes. After that, we look at the structure of the teeth, where you can see a crown, neck, and the root. So you must have heard about root canal, right? So it is the case where it is performed in cases where some individuals, they have some sort of cavity which is formed in their teeth as a result of poor lifestyle habits, maybe some genetic predisposition as well, lack of proper brushing, or many other reasons. Basically, the sugar that is present, that is staying in the teeth for or among teeth for an extended period of time, it facilitates bacterial growth, and as a result of that, it is leading degrade it degrades. So in those cases, such teeth are usually removed, and they filled in, and or they offline. But yeah, so the outermost layer is the enamel, which is like probably the strongest material within the human body, or strongest part of the human body. Then we have the dentine, the gum, is which is a part of the hard palate actually. Then the pulp, the layer of odinous lectures. You can go back to those notes if you need. So that was about the entire buccal cavity. From the buccal cavity, the next step is to move towards the esophagus, which if you've heard about it in common terms, is called as the food pipe. And the name, it's actually coined quite literally. So you have the food pipe or the gullet, which is approximately 25 centimeters long. Basically, it is the entire tract. It is a long part or elongated part of the entire tract through which food is pushed during the process of digestion. So it carries food from the mouth to the stomach. That is its main job. It is sort of the bridge between these two extremely relevant organs. And this process or this execution is mediated by a very, very important process known as peristalsis, which is an involuntary muscle movement. You don't consciously make the decision that, okay, it's time to passage some food from the mouth to the stomach. That's not how it happens, at least in case of humans. What we need to do is that we need to immediately send food down the alimentary tract, in particular through the esophagus, so that it can reach the stomach, and so that it can be broken down into its component materials, and then those nutrients can be circulated throughout the body. So this movement or this motion is basically what we understand by the name peristalsis. So you have longitudinal muscles which are present, as is shown in the lecture as well. These are present in the lower portion of the esophagus. So when these longitudinal muscles are contracting, it pushes the walls of the esophagus outwards, so that it is now catching or receiving the bolus. The circular muscles at that they relax. These contract constantly repeat. They are short, and these are repeated throughout the in the form of a wave, so that the food can actually be conducted. So the cardiac sphincter, it lies between the esophagus and stomach, and it allows conduction of the bolus into the stomach, and then the sphincter closes, and this entire extremely relevant movement or extremely significant movement of peristalsis is carried out or is finely regulated by the medulla oblongata in the hindbrain. After food has been churned, it has been crushed, it has been ground, and now it has been sent through the entire food pipe or the alimentary canal. Now comes the major organ that we think about whenever we think about the process of digestion. Whenever we say indigestion, somebody has indigestion, it is majorly associated with some sort of pain or ache in the stomach or in the tummy of such an individual. But the stomach, what exactly is it doing? First of all, you need to keep in mind that it is the widest organ of the entire alimentary canal. Majorly, it has two parts. We have the body and the antrum. So body and antrum are its two parts, but if you think about it physiologically, how it is placed within an individual and what its role is, if you actually classify on the basis of that, then first you would have the oral portion, which is the first of the body. Second means the portion that is towards the tail. That is where the word coda and caudal come from. So as food is entering in the oral portion of the stomach, it is forming some concentric circles, travels from stomach to the brain, brain back to stomach. As a result of this, the tone of the muscular wall of the stomach body reduces, and the wall sort of starts to bulge outside, so that more and more food quantities could be accommodated in the upcoming future. If you consider average capacity, let's put it that way, it majorly is around 0.8 to 1.5 liters of food. And once you have mixed these food with the stomach secretion, then digestion takes place. Also, is intrinsic factor that is required for absorbing some specific nutrients, in particular for vitamin B12 to be absorbed through the intestinal wall. So this is its specific and very narrowed down function. So that was lecture one. Secondly, we'll come to the small intestine. We have talked about the buccal cavity, the esophagus, and the stomach. And now we are moving on to the small intestine. So I'll just take a very short break before I So in lecture two, what was discussed was about the was continuing with the theme of the digestive system anatomy. So we are repeatedly saying that you are consuming food in order energy, right? So how are you getting to that point? How are you acquiring nutrients? The reason behind that is are these intestines, because they are absorbing nutrients from whatever food items we are taking in, and they are using that in order to derive nutrients, in order to further break them down to derive carbohydrates, vitamins, minerals, etc. from each of these things, which you would regularly require for each of your survival essential processes. So they perform this extremely crucial and extremely relevant process of absorbing nutrients from the food that is consumed. So if you first talk about the small intestine, then it has three parts, right? It has the duodenum, the jejunum, and the ileum. These are one out. It's a continuous organ, but yes, you can actually segregate it into these three parts. So this length, we have often learned in our childhoods as well. What is the longest organ in the human body? It is the small intestine, because of the extensive coiling that occurs in the small intestine or within the small intestine. It is approximately 6 meters, or rather 6.25 meters long. It is correlated with the height of the individual, and it is the longest part of the alimentary tract. So it has intensive coiling inside the body, so that it can be accommodated within a small space. And the chyme is conducted through the small intestine by the movement that we have discussed previously. So initially in the mouth, what was the same movement, which was peristalsis. And during this process, when it's a very slow movement. You don't want it to just get flushed, right? Whatever food you're taking it, you don't want it to be just excreted out of the body. You want absorption to occur, which is why there are so many infoldings in the small intestine, so that the entire process can be really slow. It can be gradual, and simultaneous absorption of all of these requisite nutrients can also be carried out very smoothly. So during this process only, you get complete digestion of A, proteins, B, carbohydrates, three, fats, and four, nucleic acids. So it's an extremely crucial process. So breakdown is in the stomach. Stomach is not really involved in absorption of any nutrients. These are the intestines, or this is the function of majorly the intestine, particularly the small intestine. After that, we have the next organ, which is the large intestine. And here, the reason, even though we are saying that small intestine is the larger organ of the body, largest organ of the body, um, skin is actually the largest organ. We can call it the longest organ of the body. But despite that, this is called the large intestine. Now, the question might be why. So the reason behind this could be that it has a not could be, it is actually the reason for that nomenclature, because its diameter, the area through which the chyme or the digested food is now passing through, is much larger. The cross section is much wider, hence giving rise to the name. So large intestine as well can be divided into multiple parts. We can see that it is divided into A, the cecum, the colon, and the rectum. So if you look at the diagram which is shown here, you will see that it's this terminal end, it is called cecum, and it is ending with the vermiform appendix. So cecum, you can think of it much like a pouch or a sac, and from the cecum there is a sort of an outgrowth which is called which is a vestigial. When Okay, what do we understand by vestigial? Vestigial is something which is not a necessary organ. It's some sort of a remnant of a previously complete organ that has been retained in individuals over over the course of evolution as well. It is retained within people, and it is not really performing any function. It can be removed, and it would not significantly affect the life of such people. So such organs are vermiform or vestigial. So the cecum is, however, much more developed in case of herbivores than in our case or in case of carnivores. Um, Now we come to the idea of valves. So you might have heard of valves when we talk about the heart, right, within the circulatory system. What it exactly does is it restricts or it controls the direction of flow. So from which direction to which direction some fluid, some food component, or something or the other will go is actually determined by the valves. So valves prevent the reverse transport or reverse or retrograde movement of each of these materials. So that is their function. However, when we are talking about these, in case of the intestine, what they are doing is they are preventing the backflow of fecal contents. So you can understand the significance of such a process. If such valves were not present, toxic materials which are going through so much of processing, or which are going through so many regulatory steps in order to ensure that it does not get retained in the body and it is removed from the system. If such things could flow right back into the individual's normal life or normal functioning, then that would severely affect the quality of life of such people. And therefore, this control needs to be very well maintained. And what we can see here is that the cecum, the ileum and the cecum. So we talked about the ileum, like here in the small intestine, we talked about the duodenum, jejunum, and ileum. So ileum is the here, if you look at this, the ileum is the terminal part of the small intestine, and from there we are moving towards the cecum, which is the initial part of the large intestine. So the valve, there is a valve that operates between these two to in order to ensure that there is the digested food only moves in this direction, and it prevents this backflow from occurring. And it is capable of resisting back pressure of at least 50 to 60 centimeters of water. So it is an extremely capable valve. Further, we see that after the cecum, we have the colon. So the colon is majorly, it can be divided into three parts. If you think about it much like a triangle, you have the ascending colon, because if you consider a human being as a triangle, through the middle, and then descending colon, which is terminating with a descending, and then sigmoid colon because it is a little curved like an S, and then it terminates in the rectum, and finally the anus. So the lower portion of the function of the colon is also manifolds. So while the initial or proximal parts mainly are involved in absorption and in storage, the distal parts are majorly involved in storage, particularly length of the large intestine in particular is very relevant. You can see it's approximately 20 centimeter longs, but it terminates with a smaller tract, which is the anal tract. So when you have a mass movement of feces into the rectum, when an abundant amount of feces are accumulated or gathered in the rectum, then there is a then you again have the neuromuscular system which must come in order to signal to the individual, to that living being, that the person needs to defecate now. And there is a so that's what you need to understand, that none of the um different modules that we will study in this course, none of them work in isolation. Each of them is coordinating, or there is some sort of a crosstalk which is happening here, which is regulating a human being's life. So that is what you see here. So the anal tract, it opens obviously to the anus, which has two different sphincters, and this is the most crucial step. Just like we talked about valves, right? There are two valves which operate here, the internal and external anal sphincter, because here also that very significant point of control where it determines that no fecal content should come back into the individual's body or into the individual's digestive system. That is taken care of by these two sphincters. And apart from that, apart from these absorption processes, what it also does is that vitamins get absorbed in the by the bacteria, which are residents of the large intestine. And majorly, you can see that vitamin B complex and vitamin K are are absorbed here. So they perform this extremely important function as well. So here we conclude with the large intestine, and from this point onward, we will move to the second set of um, or the second system that operates in the process of digestion. So if you remember, we had initially said that there is a physical or mechanical aspect of it, starting with the buccal cavity, the esophagus, the stomach, small and large intestine. Now we come to the second set, which are the glands, which are not directly doing anything, but their products or their um, components, what they are contributing to the digestive process, they are performing the extremely relevant process. And again, either of these two will not be able to function in isolation, and you need contributions from both of them in order to regulate or control the entire process of digestion and make it fruitful. So we'll start with the glands now. So first we are looking at the secretory glands. So what you understand by the name secretory, it means it is releasing or it is secreting um, some component or some, you can directly understand that it would have to secrete some enzymes, particularly digestive enzymes, which are helping in the process of digestion of food. But it is not just limited to that. It is also secreting mucus. Why mucus? We'll come to it in great detail. It is also an extremely critical requisite in ensuring that digestion process is smooth within an individual. And if there is some problem in the mucosal lining or in the mucus lining of a person, then it severely affects their health and it can cause a lot of disorders. So that is also something that we need to look into. So we are first looking at salivary glands. So the major gland of salivation. So first we're looking at the glands that operate in the mouth. If you're going by the sequence of organs that are a part of the digestive system as well. So first we're looking at the salivary glands. So as the name indicates, they are involved in producing saliva. So major glands that operate here are the parotid, submandibular and sublingual glands. So if you look at the diagrammatic representation here, you'll be able to see the submandibular, the sublingual gland, and also the parotid gland, which is much closer to the ear. But yes, it works in concordance with these other glands in order to stimulate the release of saliva. So apart from that, there are other small buccal glands as well, as you can see, Stenson's gland, Ravenian duct, Warsman's duct, etc. So a healthy individual is capable of releasing around 0.8 to 1.5 liters of saliva daily, which is a large amount. So what it majorly is made up of are two proteins. The first is tyalin. Tyalin is a form of alpha amylase, which you also commonly know as salivary amylase, the first enzyme, which is why the digestion of carbohydrates, it's said to begin in the mouth or in the buccal cavity. We'll come to this a little bit later as well, where it is mentioned in much greater detail. But yeah, these are the first component, or this is the first component, which is tyalin. Apart from that, you also have mucin, which is released, and mucin is involved in majorly in protective functions. So like we said, mucus is a very relevant, very important part of the entire procedure. So what it does is mucin provides a protective layer within the entire alimentary canal, but in particular, since we are just talking about salivary glands, they are involved in binding of the bolus together. So sorry, parotid glands are majorly involved in tyalin release, whereas the submandibular lingual glands, they secrete tyalin as well as mucin. And the other small glands that we talked about, they only release mucus. pH is maintained between 6 to 7, and esophageal glands are only releasing, secreting mucus, which is providing the entire covering or coating, or what you commonly understand as lubrication, for allowing the process of swallowing. So what you have chewed up in your mouth, that needs to be gulped, or that needs to be swallowed to allow its entry into the esophagus, and then its passage through the entire tract. So for that, mucus lining, or mucin particularly, is extremely important, and esophageal glands are a major contributor to that. The next set of glands now present in the stomach are more inclined towards the process of breakdown, or towards the process of stomach digestion. So these are commonly called as the gastric gland. So here you can see a diagram of the stomach, and this is a gastric gland, which is made up of a number of different types of cells. It's not a lining of only one type of cells. These are different. You can think of them as a heterogeneous group of cells that are coordinating and acting in a united, or you can say a coordinated manner, in order to allow proper food digestion. So you'll see, so you have the entire surface of the stomach, which has cells that are constantly releasing mucin under normal circumstances, in order to maintain that smoothness, or in order to maintain that easy flow or easy passage of food items or digested chyme through the entire stomach. So apart from that, the stomach mucosa, which is the stomach lining, that has two types of tubular glands. We have the oxyntic cells or oxyntic glands, and the pyloric glands or pyloric cells. What each of them does is they have distinct functions. So the oxyntic glands are involved in HCl secretion, pepsinogen, intrinsic factor, and mucus, whereas the pyloric, other set of cells, are majorly involved in releasing mucus, which is the reason why it is providing some sort of protection from the acidic contents of the stomach. And they also release gastrin hormones. So what you need to do is you need to separately, for your own better understanding and for easy recollection, you make a table or a chart with each of these glands, what they are secreting, and what is the function, and is it just a protein, is it just an enzyme, is it a hormone. You write each and every one of these, because these are going to be helpful for an indefinite period of time if you decide to stay in the field of biology. So it's for better clarity, better understanding. You can try listing these out and forming a sort of chart for better, easier, and more productive way of studying. So next we are looking at the liver. The next set of glands are located in the liver. So the liver, you can think of it as the largest gland in the body. It releases an insane amount of bile, about 0.6 to 1 liter, like an entire bottle of bile is released per day. What does bile do? Why are we bothered with it? So bile is doing two things. One, it is digesting fats, and it is helping in absorption. So bile acids in particular are the reason why bile is able to emulsify the large fat particles into many small particles. And what it is doing is, you have fats. When you're emulsifying fats, you're increasing the surface area where digestion can take place. And the surface of these small particles can be attacked by lipase enzymes, which are secreted in the pancreatic juice. So if you look here carefully, you'll see that this is where the stomach lies, and much larger to it, but lying in very close conjunction, is the liver and also the pancreas. So the pancreas and the liver are sort of coordinating in the process of fat digestion. So while the pancreatic juice has enzymes which are commonly known as lipases, and as the name indicates, it is involved in breaking down lipids or digesting lipids. These lipids, or the process of lipid digestion, it begins once the liver has emulsified the fat. So the reason why you'll see majorly the grown-up members of your family, going through so many tests and checkups every year is to make sure that each of these organs is functioning properly. Because, like you can see, they're all together performing some function or the other, and therefore their coordinated or synchronized proper functioning is crucial for their better health in the long run. So that is something you should also be mindful of. Bile acids, they aid in the absorption of the end product digested fat through the intestinal mucosal membrane. So the end product that is produced, bile acids are helping in that digestion, and that is occurring through the intestinal mucosa. Also, the liver is very, very important in removing waste products from blood. So, one example is the product bilirubin. So, what is or where is bilirubin at all being produced? It is an end product of the process of hemoglobin digestion, and also the excess of cholesterol that might be produced is also excreted out with the help of bile. And you can see that a pear-shaped structure that is attached to the posterior surface of the liver, which is the gallbladder basically, that can store 30 to 60 ml of the bile that is secreted by the liver. And you've heard about the gallbladder, if not in any other context, majorly because people are said to get stones, which are basically salt deposits, which are formed within the gallbladder. So it is the storehouse of bile, basically, and therefore it also has a major role to play in the process of digestion. Now coming to the next organ, which operates in concordance with the function of the liver, is the pancreas. So the pancreas is a soft, lobulated, large compound gland. We'll come to this term, whose internal structure is similar to the salivary gland. Why it is called a compound gland is because it has um, a consolidated assembly of many different types of cells. You'll learn about it in detail as well. You have alpha cells, beta cells, and each of these cells have different functions to play. But what we are concerned with is the fact that pancreatic acini, which are acinar cells, they secrete the digestive enzymes, whereas a large amount of sodium bicarbonate solution, a base, is secreted by small ducts or ductules and larger ducts. These mixtures of enzymes along with the base sodium bicarbonate passes through the pancreatic duct. Pancreatic duct, it joins with the hepatic duct. Hepatic duct is coming from the liver. So if you look here, let me just show you. This is the pancreas. This is the pancreatic duct. This is the common bile duct from the gallbladder, and you have the hepatic duct from the liver. All together, they are joining. They're joining together, and they are forming. They're together emptying into the duodenum through the papilla of water, so which is here, and from there it will enter whatever fat digestion etc has happened, that will open up into the small intestine for further digestion to take place. In the pancreatic enzyme or in the pancreatic secretion, we have enzymes that are capable of digesting all three of the major food components, which are carbs, proteins, and fats. So if you look at the enzymes, you will find a lot of familiar names like trypsin, chymotrypsin, carboxypeptidase, all of which digest proteins, and that is actually the context in which we have learned them as well as proteases. But also you have pancreatic amylase. You had salivary amylase here, otherwise known as ptyalin, but also pancreatic amylase is present, and this is released by the pancreas, and what it does is it uses carbohydrates as the substrate and breaks it down. And apart from that, we also have pancreatic lipase, which is one of the enzymes we had talked about. We have cholesterol esterase and we have phospholipase. Together, they help in digestion of fats. Finally, we are coming towards the intestinal glands. So we talked about the glands in the mouth. We talked about those in the stomach, and those in the small intestine are our next. But before that, we also talked about the pancreatic ducts as well, or pancreatic glands as well. So these are formed, intestinal glands are formed because the surface epithelium of the small intestine, it sort of modifies. What you get are Brunner's glands and crypts of Lieberkühn. So Brunner's glands are more sparsely located, one might say. They're only found in the initial part of the duodenum. What they are involved in is secretion of a very large amount of mucus, alkaline mucus, which protects the small intestine wall, or in particular the wall of the duodenum, from highly acidic gastric juice, and it also neutralizes HCl, or hydrochloric acid, that is coming from the stomach. That was the Brunner's gland. The second set is the are the crypts of Lieberkuhn, which are small pits. When we say pits, they basically mean, if you think about the surface of the small intestine, it has a lot of villi and microvilli. We'll come to that. But the lower portion, the little curvature at the interior, is basically what we understand by crypts of Lieberkuhn, which are small pits located all over the surface of the small intestine. They lie between these villi, between the intestinal villi, and they are covered by the epithelium. Majorly, they consist of two types of cells. So we have goblet cells, which secrete the mucus, and they are the major contributors. But we also have enterocytes. The word entero usually means intestinal. So enteroendocrine cells are those which are present in the gut that are releasing certain enzymes. So, or they're releasing some sort of they're secreting some sort of an enzyme. So, or hormone, particularly enteroendocrine basically means that. So, enterocytes in particular are involved in secreting water, electrolytes, and along with the end product of digestion over the villi surface. At the base of these crypts, if you see, this is an entire crypt, and at its base, what you see are those enteroendocrine cells, like I just talked about them. But what you see here are Paneth cells, as is indicated here. You have argentaphin cells that are also present. So what you see here is an example of a compound structure as well, which is what we saw here. This was a compound gland, a separate gland. Here you see a number of different types of cells all together present at the crypt in order to perform some specific functions. So these are some of the enzymes that are secreted in the entire alimentary canal. Like for example, tyalin, which is helping in starch digestion in the mouth, pepsin, gastric lipase, renin from the stomach. In mostly children's stomach, you will find casein. Lipase is for fat digestion. Pepsin is in the stomach for digesting proteins. Pancreatic amylase digests starch. Trypsin, chymotrypsin, elastase majorly digests proteins. In particular, elastase is specific for elastin. Then we have carboxypeptidase for large peptides. Pancreatic lipase, as the name indicates, will digest lipids or fats in particular, here with specific inclination towards triglycerides. Then nuclease, which helps in digesting nucleic acids, DNA and RNA. Small intestine is the location of nuclease as well. Then we have enterokinase, which digests trypsinogen, aminopeptidase for large peptides, dipeptidase, disaccharide, intestinal lipase, nucleotidase, and nucleosidase. So you have a large range or variety in the type of enzymes which operate, and based on that, they would perform their individual function. In the third set of, or in the third lecture, what basically was done is, after an initial recollection, we look at each of these types or subtypes of digestion in isolation. So starting with the process of protein digestion. So if you consider any food material which you consider or which is marketed or known to be protein rich, what happens to that substance? You just don't get protein out of nowhere, right? What happens is that, first of all, in the stomach, some proenzymes are present. What do we mean by proenzymes? Enzymes which are localized or which are present in their like inactive form. Those are proenzymes. So the reason why these need to be kept in the proenzymatic condition is because you don't want cells to be constantly or you don't want everything in the cell, everything in the stomach or everything in a system to get completely digested. You want only food items which are getting activated under certain conditions at certain point of times to carry out the process of digestion. So for that reason, under normal circumstances, we would want these enzymes to be present in their inactive or partially active or suppressed form. So that's what we see here. Two proenzymes are present that are pepsinogen and prorenin. In particular, renin is present in the stomach of little children, little babies, whereas proenzyme pepsinogen is present in adult stomach as well. So that is what we see here, that pepsinogen, it gets digested by HCl and by pepsin. HCl is coming from the stomach, obviously. What it is giving us is pepsin. The active form of the enzyme will come to its next function in the next slide. And prorenin, which was initially present in the inactive format, which gets activated, is producing renin. And again, this is only for children because of the difference in their diet, because babies or children, they do not feed on the type of food that we eat. They have more restricted intake, and based on that, their differences can also come in. So yeah, so pepsin and renin are produced. Now what happens is that renin in particular, like you were just saying, is majorly found in the stomach of children or a small child. The reason for this is because their diet is heavily dependent upon consumption of milk, and milk is very rich in casein, a particular protein, casein, which we commonly know to be the milk protein as well. So casein gets digested by renin, and what is produced as a result is paracasein. So paracasein, along with the calcium which is coming from milk, is producing calcium paracaseinate, a sort of digested product, and this is the end product which you get from renin digestion. Pepsin is acting as an enzyme that is going to digest a large number of proteins, and it is going to give you smaller products like peptides, etc. But we'll get into more detail about that in the small intestine. So if you see in the next step, in the small intestine, you will find majorly three proenzymes. First is trypsinogen, second is chymotrypsinogen, and third is procarboxypeptidase. So first we see trypsinogen, which is getting digested by enterokinase. Enterokinase, like I said, entero is usually indicative of some component within the intestine, and that along with trypsin is producing more and more trypsin. So trypsinogen is from the inactive form is producing trypsin, the active form, and chymotrypsinogen is getting digested by trypsin to produce chymotrypsin, which is the active form of the enzyme again. And procarboxypeptidase also gets digested to produce the active form, which is carboxypeptidase. Now within the duodenum, further digestion is occurring where protein and peptones. So peptones get produced in the stomach itself. Like we said, digestion by the pepsin, which was present in the stomach, is often going to lead to the formation of peptones. Majorly, peptones are found in animals, or you can think of it as individuals that have an animal-based diet usually are found to be much rich in peptones. And proteins and peptones are getting digested by trypsin to produce peptides. And secondly, we have casein, which is getting digested by chymotrypsin to produce paracasein. And paracasein, along with calcium, is producing calcium paracaseinate that we previously discussed. And calcium paracaseinate is getting digested in the small intestine to produce peptides and calcium. And this digestion is being performed by chymotrypsin and trypsin that are present, or that are intestinal digestive enzymes. Finally, we are looking at some other reactions as well, which is centered around elastin. So elastin is majorly component of those, or is one of the proteins or one of the components that need to be digested in individuals who have an animal meat-based diet, not usually for plant-based diet eaters, so consumers. So elastin, along with water by the enzyme elastase, is used to digest elastin in order to produce peptides, and large peptides are digested by carboxypeptidase in the small intestine to produce smaller peptides. So trypsin, chymotrypsin, carboxypeptidase, elastase, these are all enzymes. Usually not elastase, but the rest, the remaining three are extensively used in the research in research labs as well for digestion of proteins and for trypsin is extensively used in cell culture. You require trypsin to even detach cells from a culture dish because it is digesting the matrix metalloproteins and it is um, sort of the solution to adherence. So it's adhered cells would detach when you treat them with trypsin. So that's how it is functioning. And in the jejunum, peptones and small peptides are getting digested by aminopeptidase to produce oligopeptides and amino acids, following which carbohydrate digestion was also looked at. I'll wrap this up a little faster because the discussion has exceeded the initially estimated time. So you can see in carbohydrate digestion, starch is getting digested in the mouth by ptyalin, salivary amylase, maltose, isomaltose, and dextrin is produced. The rest of the digestion you see of carbohydrates is occurring in the small intestine of each of these individual components to produce what? To produce glucose and galactose, simpler carbs, or in fact, you can think of them as simple sugars. The reason why this is necessary is because you cannot, under most circumstances, you would not be able to use a carbohydrate in any metabolic pathway to produce energy. You would require digestion in order to produce glucose, which can enter glycolysis and can go through the Krebs cycle, and then it is going to help you in production of energy. So the food that was taken can actually be translated to energy by this process, apart from the fact that you also need to absorb nutrients and acquire energy from and help in other processes as well. So gut bacteria that are resident gut bacteria or microflora, as we commonly know them, they are capable, when they are present, they are capable of using lactose that we get from milk and from several other sources. And their process of respiration often produces a lot of gas. So people who are lactose intolerant, they usually lack the enzyme lactase and hence are not able to digest it, and they suffer from a lot of flatulence and a lot of indigestion and other lifestyle problems as well. So finally, we come to fat digestion, which here you see is the backbone. You have glycerol and 3 fatty acids together, which forms the triglyceride, and that undergoes the process of digestion. So this is a summary of sort of a graphical summary of the entire process of digestion of each of these different components. Nucleotides have not really been discussed in much detail because it's not that extensively performed. But yeah, basically, if you have a polymer, majorly a polynucleotide that gets digested to get individual nucleotides, nucleosides, nitrogen bases, and finally pentose sugars, which can enter other pathways and be metabolized by the individual who is consuming them as well. And finally, we come to villi and microvilli. So these are extensively present along the wall of the intestine, the small intestine. They are often also called brush border epithelial cells because they are tiny projections along the entire surface, which gives an appearance of it looking much like a brush, like tiny hair-like projections, which look like they are surface of a brush. And the reason they are present is because they increase the surface area for absorption within the intestine by leaps and bounds. There is a huge difference in the surface area that was, if you consider a flat surface versus if you consider a surface which has an immense number of projections, then each villi is providing some extra surface area from which absorption has to take place. So it is increasing the efficiency of this process of absorption, and it is making sure that only those products which are not of any value to the individual who is consuming them, only those are removed, and everything that can be of use or that is essential is getting retained within the individual's body and their circulation, and they can be utilized for other purposes. And here is, that is what you see. You can see that they are innervated. You have blood capillaries, venules, the connective tissue which is present around them. And here is the process of absorption. I'm not going into details of it. Each of them gets absorbed across by transporters by these brush-bordered epithelial cells from the luminal side towards the blood capillary into the blood, and that's where the circulatory system comes in carrying it to each of the um, requisite destinations or accurate destinations, let's call it that way, and then they can execute their individual functions. And finally, we look at the absorption of water as well, which is performed by these enterocytes. And an example of all of the diseases that could result if these processes are not occurring the way they're intended to. So you have jaundice, vomiting, diarrhea, constipation, marmus, kwashiorkor, rickets, osteomalacia, diabetes, acid reflux, constipation, etc. So each of them result from different defects. And with that, we kind of completed rather extensive recap of this week's topics. But yeah, we'll go through the questions to wrap up this week's interaction. So let me just put it on slideshow. Yeah, so we have done the week 8 recap in quite a lot of detail. Now coming to the assignment questions, the first question was about organisms that fulfill the requirement of food from rotten parts of dead and decaying matter are known as a holozoic organisms, the saprophytic organisms, which, as we said, they release or they secrete some digestive enzymes outside of their body, and whatever material is present on the outside that undergoes digestion because of that is broken down by these released enzymes, and then the digested material is taken up after simplifying the process of digestion. Saprophytic organisms or saprophytes are able to take up those nutrients in a much simpler manner, much quicker manner, and it is sort of an adaptive strategy for these types of organisms. Then coming to the question number 2, which was about tapeworms. What is the mode of nutrition that they follow? So obviously, if you've heard of people who suffer from tapeworm infections, you would know that basically they are parasites, right? They majorly reside within the intestine of individuals. If they have a tapeworm infection, majorly they are found to be present in meat, some sort of meat. So if a person is consuming incompletely or improperly cooked meat, they usually suffer from some sort of a tapeworm infection under a lot cases, because their eggs host body do not provide any help. There's no symbiotic association which is occurring there. They are just taking up whatever lights. Next, coming to pepsin, it is predominantly present in A, stomach, B, large intestine, C, small intestine, or D, the mouth. So pepsin, we have discussed this extensively. Pepsin is restricted to the stomach, and it is involved in the digestion of proteins from pepsinogen, the proenzyme, to pepsin is produced, and then pepsin is involved in digestion of milk proteins and other proteins as well. It's breaking them down into peptones and peptides, smaller peptides, and eventually those get digested to amino acids, which would enter different metabolic pathways in order to help the survival of organisms, and also they add to the amino acid pool. That is why you are always, not any individual is always fed to consume more and more proteins, because that will give you the amino acid pool apart from other sources, apart from de novo synthesis. When you supply more amino acids to your body, when your body needs to synthesize new amino acids, it can charge the RNA, like we discussed during the process of translation. It can charge the RNA from that amino acid pool and then generate more and more new polypeptides and new proteins and allow that organism to survive. Then coming to question 4, parotid glands are present: A, near the ears; B, near the liver; C, near the esophagus; or D, at the center of the brain. So if you remember that diagram that we had initially seen, parotid glands are located somewhere here, close to the ears, and along with the sublingual and the submandibular glands together, they are helping in the process of digestion. These are the glands that are secreting some sort of either they're secreting tylen or they're secreting mucin, which is helping in the process of digestion in some way. Either it is breaking down carbohydrates or it is helping in binding the bolus some way or the other. In question 5, we have been asked that the mixture of food and saliva that is formed in the mouth after chewing is known as. I just answered this question. So here we have A, vestibule; B, chyme; C, bolus; or D, uvula. So vestibule is obviously the wrong answer. We discussed that vestibule. In fact, it was one of the sample problems discussed in the last week session as well. So vestibule is the gap area between the cheek and the palate, or the like the area between the lip, cheek, and the palate. So basically the gap area here, those are the vestibules. Uvula is the part at the back of the throat. If you have ever seen a sort of a little dangling portion at the back of the palate, upper palate, is the uvula. However, chyme and bolus are very similar, but it is about the location. So if you've been asked in about the entity that is formed or the mixture that is formed in the mouth because of the process of chewing, then that would be the bolus. But when the bolus passes through the alimentary tract and it has gone towards the stomach, at that point it is the chyme, because it has undergone some sort of digestion and it is now, its composition is different. So it is the chyme or the chyme. In question 6, we were asked which of the following is not a part of the tongue: the foramen cecum, the follate papillae, or the sulcus terminalis. If you refer to the diagram of the tongue, then you would remember that, and option D is uvula. You would remember that option D is not a part of the tongue. Like I just said, it's part of the palate. It's a continuous part of the palate when you are transitioning towards the esophagus, towards the food pipe or the digestive tract, basically. On the other hand, the other three are actually located on the tongue surface. The sulcus terminalis sort of divides the tongue, and then you have the follate papillae, different types of papillae, which are involved in tasting or the sensation of different, or the distinction between different types of taste. And foramen cecum is also involved in a similar function. Then we are coming to the dental formulae. So the number of teeth in adult rabbit, humans, and dogs are dashed respectively. A very direct question. Obviously, most of us would know about humans. We have 32 teeth, but unfortunately, all of the options have 32 as an option. So you would need to remember it. These were given as examples. So that would be for adult rabbits, it is 28, and for dogs, there are 42 teeth. So this is quite a very memory-based question. The gullet is also known as, if you remember the discussion about each of the different parts of the digestive system, then we have gone through alternative names for the food pipe. For the alimentary canal or esophagus, where it is also called as the food pipe, and it is also called as the gullet. So very subtle questions, but yeah, you need to go through the lecture notes. If not individual lectures, you must go back to the lecture notes and try and keep note of these subtle, what should I say, anecdotes, or subtle alternative names, or question, potential questions that can come up from these areas. So yeah, that's the food pipe. The ileocecal valve, it guards. I'm not reading out all of the options because this is quite a direct question. Basically, the ileum and the cecum are spatially kept separate, or the contents of the ileum and the cecum are spatially kept separate so that fecal contents do not flow back between the two of them. So after the ileum has ended, that is the small intestine has ended, to the part where the large intestine has started, you do not want any backflow of components from from the latter to the former, and that is taken care of by the ileocecal valve. Then in question 10, we were asked that myelin is secreted in A, the stomach, B, the small intestine, C, the large intestine, or D, the mouth. So myelin, if you remember, is basically salivary amylase, and it is released in the mouth, not the stomach, not the small or the large intestine. Then coming to question 11, kwashiorkor is caused due to A, protein deficiency, B, water deficiency, C, carb deficiency, or D, lipid deficiency. So you saw a list of diseases. You would have to go back and go through each of them. But yeah, in particular, like marmas and kwashiorkor were specifically mentioned. So kwashiorkor particularly is involved in or is a result of protein deficiency. The substrate for renin, renin, as we have discussed previously, uh, is an enzyme or is a digestive enzyme that is majorly present in children or in babies. And the reason for that is the type of diet that they are majorly consuming, which is heavily dependent on milk, or is a milk-based diet basically. So casein is the major milk protein, and therefore renin is involved in its digestion, and then calcium comes in and it helps in calcium paracaseinate formation. So that's the entire process of digestion that we have also discussed. Coming to the last three questions for this week's assignment were in question 13, you see that the incisor teeth are used for A, tearing meat, B, cutting food, crushing food, or grinding food. So each of these functions are attributed to different types of teeth. In particular, for incisors, like we said, the front two teeth are involved in biting something or cutting some sort of food. So like I gave the example of an apple, right? So they are involved in cutting food. Meat cutting or meat tearing in particular, because it's very elastin rich, is taken care of by the incisors, which are more pointed teeth, which are able to do the same. Crushing of food is taken care of by the molars, and those food are further grinded by the premolars. So molar and premolar are involved in further breakdown of the food, but yeah, incisors are involved in cutting. In 14, we're being asked that human beings exhibit dash mode of nutrition. So we exhibit a holozoic mode of nutrition because we are consuming parts of plants or animals directly and then breaking them down. And finally, in question 15, we were asked that the pH of the stomach is acidic because of the presence of lactic acid, hydrochloric acid or HCl, sulfuric acid, or uric acid. Very, very direct question. The major contributor of the process of digestion, particularly when we talk about the stomach, is HCl. And it is the reason why the pH of the stomach is acidic as well. Now, we are quickly going to go through some sample problems for week 9. We will discuss these topics in the next live interaction session. So I will not be going into details regarding these, but yeah, for reference, for some idea, you can go through these questions. It might be helpful in solving the assignment for this week as well. So first question is about, because this majorly is about the circulatory system. So first we are saying question 1 is about how the heart pumps the fluid continuously through the body is through A, small intestine, large intestine, windpipe, or blood vessels. So very naturally, blood vessels are responsible for conducting or carrying blood as well as other nutrients. Apart from nutrients, let's stick to just the circulatory system. So from the heart, the double circulation system, which is operating, which is circulating blood throughout the body to each and every organ, supplying oxygen to them and carrying away any waste and deoxygenated blood from those areas. All of that is done by blood vessels, arteries, and veins. In question 2, we have been asked that the type of circulatory system that is seen in earthworms is A, open circulatory, B, closed circulatory, C, no circulatory system, or D, both. So the option here or the answer here is the closed circulatory system. I'm sure if you've gone through the lectures, you already understand the distinction, but yeah, we'll discuss this these subtypes in more detail in the next week's interaction session. In question 3, we were asked that the deoxygenated blood in fish is converted or is turned into oxygenated blood by A, heart, B, lungs, C, gills, or D, blood vessels? So the answer here is gills. Fish, the circulatory system, if you might say so, in case of fish, is majorly operated via gills. So this deoxygenated oxygenated blood exchange is executed by gills in case of fish. Uh, However, in case of frogs, these two types of blood get mixed in which part of their body. So you have option here as ventricle, right atrium, left atrium, or both atria. So the correct option here is the ventricle. So that is where this mixing is actually occurring, and that's what you see in the ventricle. Then in question 5, we are being asked that they have thin walls located, like which component has thin walls that is located closer to the surface of the body, and which also have valves which prevent black backflow and which ensures that blood is always flowing in the right direction. Is A, capillaries, B, arteries, C, veins, or D, arterioles? Sorry. So each of these components correspond to veins, a subtype of blood vessels, just like we discussed. They are thin-walled, which is why you can feel blood pulsating through them, whereas arteries are more thick-walled. They are closer to the surface of the body, which is why you can usually see like blue lining or blue bluish deoxygenated blood carrying veins on the surface of your body, and they have bulbs because they are carrying the oxygenated blood. It should not flow back to the original site. So basically you are seeing that whenever there is something in the body that is potentially harmful, that is never allowed to or that is there are mechanisms in place that keep a check on the direction in which it is flowing. So that was the case for uh, toxic fecal contents, and that is the case for deoxygenated blood, which is potentially carrying a lot of harmful things. So that's how these systems operate. Then in question 6, we are asked that the oxygen and nutrients that are exchanged for carbon dioxide and waste, where is it occurring? So if you think about arteries and veins, there are very thin walled areas where the two meet, and that those are the capillaries, and that is where you can see the exchange between the two occurring, and each of them gets carried over to their predetermined destination. And this exchange is extremely crucial because otherwise, where else would you make sure that what was harmful is taken away and what is required, which is oxygenated blood, is supplied? So that occurs at the capillaries, and we will again get into details about this in the next week. So that was question 6. Then we have in question 7, the division of atria and ventricles in a four-chambered heart is A, atria receives the blood and ventricles pump it. B, ventricles receive blood, atria pumps it. Right atria receives blood and left atria pumps it, or a right ventricle receives the blood, oops, and ventricles pump it. So the correct answer for this is about in a four-chambered heart, what you do is if you imagine the structure of the heart, the human heart, right? You have atria at the top, ventricles at the bottom. So atria, they first receive the blood. So whatever blood is coming in, whether it be right or left, is initially receiving the blood. Then it is sent via valves which are in place towards the ventricles, and from the ventricles they are pumped wherever it is required, whether it is if it is oxygenated blood via the aorta to the rest of the body, or if it is deoxygenated, it has to be sent to the lungs and then for exchange. And for heart rate for the AV node is not reading all of the options, but yeah, it's a direct question. It's 40 to 60 beats per minute. AV node is the atrioventricular node. So again, very direct answer to the question. In question 9, we're being asked that the oxygen carrying protein in blood is present in, obviously the protein is hemoglobin, and it is present in RBCs, so otherwise also known as erythrocytes. The red coloration is also attributed to the same. So that is what you see in question number 9. Then in question 10, we are being asked that the hormone that is essential for the production of red blood cells is synthesized in A, bone marrow, B, spleen, C, liver, or D, kidney. So hematopoietin, so hematopoiesis, the process is uh, regulated uh, by the kidney. You can, let's put it that way. It's the kidney, which is the answer for this. In question 11, the buildup of plaque in atherosclerosis, it occurs in A, the external lining of a vein, or B, the inner lining of a vein, or C, the inner lining of an artery, or D, the external lining of an artery. So usually arterial within the artery, where you have a, in general, blood is pumped at a, or blood flows very fast, or it is pumped under a lot of pressure. uh, Within that, within those blood vessels, when there is a buildup of plaque, what it effectively does is it shortens the diameter through which blood can be pumped. That effectively leads to higher blood pressure, which is what we commonly known as high BP, right? Which is something that individuals usually tend to suffer from when they age, because of lifestyle choices, because of food, because their body functions begin to deteriorate from their younger, more fitter days. And these are very generalized patterns seen in these kind of in this age group. So that is what you see as a consequence of age, and as a consequence of higher cholesterol, which is what you see, the buildup of plaque in inner lining of an artery, making it the correct answer. In question 12, we are saying that the normal pressure in arteries when the heart rests between beats is it is diastolic. I'm getting to the answer directly. It is diastolic and approximately 80 mercury. So 80 millimeters of mercury. So that's the direct answer to this question. Um, then coming to the immunity-based questions, the cells that activate TC cells or T cytotoxic cells to kill infected target cells in adaptive immunity is A, regulatory cells, B, helper T cells, C, memory T cells, or CD8 cells. So TC cells get activated by helper T cells. So if you remember, we had said that there is a crosstalk between humoral and cell-mediated immunity, and T8 cells are the reason why that crosstalk happens. Again, we'll get into this once again in the next week. In question 14, we are being asked that the myelin sheath of neurons that rotates around the axon for insulation is A, Schwann cell, B, astrocyte, C, microglia, or D, satellite cells. These are Schwann cells. Direct question. Then coming to 15, the homodimeric protein of the sarcomeric Z line that anchors the end of F-actin to uh, to it is A, desmin, B, alpha-actinin, C, troponin, or D, myosin. The answer is alpha-actinin. We'll get into the details about the sarcomere as well. So the striated appearance of a skeletal muscle tissue is due to the repeating bands of the protein, actin, and myosin. Again, this is the sarcomere associated question. The type of contraction that occurs during the lowering phase of a bicep curl is eccentric contraction, isometric contraction, isotonic, or twitch contraction. It is an eccentric contraction. We'll get into details about it next week. The neurotransmitter upon binding that leads to the influx of sodium inside the cell during an action potential of the muscle fiber is epinephrine, serotonin, acetylcholine, or dopamine. It is acetylcholine is the correct answer here. In 19, we're being asked that the muscle tissue which shows cross-linked stripes is A, skeletal, B, cardiac, C, smooth muscle, or D, visceral muscle. So cross-linked stripe in the striated muscles are because of or are located specifically in the heart. So those are cardiac muscles. And finally, in question 20, we are being asked that the degenerative disorder of the central nervous system that affects muscular system and movement of the individual is A, Parkinson's, B, muscular dystrophy, C, myasthenia gravis, or D, carpal tunnel syndrome. The answer is Parkinson's disease for this question, because as you might have seen, a lot of individuals suffer from it as well. It's a debilitating disease, and there is no particular cure to it, and it very significantly affects the lifestyle of such individuals. And we'll come to more details about such disorders in the next week as well. So I will stop sharing at this point. And yeah, so for this week, we basically looked at the recap of the topics that were previously discussed in the lecture notes as well. And we went through the assignment questions, and we also discussed some sample problems for the topics that would be covered in the upcoming week. And that pretty much concludes our session for today. I hope these are helpful for you. If you want additional notes or questions, you can let us know. And yeah, so for now, that is what we are wrapping up with. We discussed only the process of digestion for this week as part of physiology, and we'll be getting into much more details about human physiology in the next week. And yeah, with this, I will end my session. So thank you. I hope these are helpful, and have a good night.